Good day, everyone. I'm Philomin Juliana from the Bulldog Institute for South Asia, and my presentation today is on data-driven line advancement and parental selection decisions in the CIMMYT bred wheat breeding program. For any successful breeding program, data-driven selection decisions play a key role in enhancing the efficiency of the breeding program, and they also provide improved breeding outcomes and help us to optimize the current breeding strategies. In the CIMMYT bred wheat breeding program, all the early generations from F1s to F5s or F6, all of them are selected and bulked. Selections from these generations lead to the small plots or the pre-yield testing stage, where there are about 18,000 lines, and all of them were genotyped in this cycle. Selections from these small plots result in the stage 1 yield trials, where there are about 8,000 lines that have both genomic and phenotypic data. And selections from the stage 1 yield trials lead to about 1,120 lines in stage 2 yield trials that also have genomic and phenotypic data. And then selections from these stage 2 trials result in about 485 lines that have both genomic and phenotypic data, and these form the South Asia bred wheat genomic prediction yield trials, and the lines are evaluated for grain yield in different sites in South Asia. Finally, we have the stage 3 bred wheat yield trials, which has about 252 selected lines that have both genomic and phenotypic data, and selections from all these stages result in the international nurseries that are distributed by CIMMYT. So in these different stages, the CIMMYT bred wheat breeding program uses a combination of genomic selection, selection indices, combining both genomic and phenotypic selection, utilizing GWAS results, and also getting some basic quantitative genetic parameters to help in selection decisions. And I'll be talking about each one of them in my slides. So the first stage here, the small plot stage or the pre-yield testing stage, where we have about 18,000 lines, so all of which have been genotyped. So we use the genomic estimated breeding values of all these lines to advance lines for different breeding pipelines. And these are for traits that are not phenotyped in the small plots. So for all these lines, 18,000 lines, we just have the genomic estimated breeding values and no phenotypes. And we've done it for several different traits for grain yield in the bed 5 irrigation, flat 5 irrigation, early heat, bed 2 irrigation, drip irrigation, and late heat environments. And we also have the GEBVs for different diseases, blast, fusarium head blight, septoria, spot blotch, stem rust, and stripe rust. And for grain zinc and iron, for some end-use quality traits like alveograph and low volume, and also for days to headache. So thanks to the USAID Feed the Future project and Jesse Poland's lab from KSU, we've been able to genotype all these lines and also use all these genomic estimated breeding values in the selection decisions. We also have different selection indices for these that make use of these GBVs. And this information is used to advance lines for different pipelines. For example, let's take the hard white optimum environment normal maturity pipeline. And there are several key traits in this pipeline, and each of those traits are having different priorities. As you can see here, for example, high and stable yield is a very high priority for this pipeline. And end-use quality, grain zinc and iron are also of high priorities. And there are some traits like wheat blast and spot blotch that are of a low priority for this pipeline. So considering all the priorities for the different traits, we use the GBVs in a selection index for different pipelines and deliver that information to the breeders to make selections. And the next stage, the stage 1 yield trials, which is about 8,000 breeding lines and selected from the small plots. So in this stage, genomic data is integrated with phenotypic selection. So we have the grain yield GEBVs, and this is also combined with the grain yield phenotypic data, and that helps us to increase the selection accuracies. And in the figures here, I'm showing you the within cycle grain yield predictions and across cycle grain yield predictions. So this within cycle grain yield predictions, the accuracy was about 0.44, and this is just within cycle 5-fold cross-validation prediction accuracies. And the across cycle grain yield prediction accuracy was about 0.33, and this accuracy is from using the previous cycle's data, grain yield data, to predict this cycle's data. And as you can see very well, we have the grain yield blues on the x-axis, the grain yield GBVs on the y-axis, And the different quadrants here are showing, and different colors here indicate lines that have not been selected by genomic selection and phenotypic selection. So you can see the red dots here. So these are lines which are not selected by both these selection methods. So this is assuming that we select the top 10% of the lines using both the methods. So about 88% of the lines can be discarded using both phenotypic and genomic selection. And about 32% of the lines were common between phenotypic selection and genomic selection. So you can select the top 32% of the best lines using within cycle predictions, and using the previous cycle predictions, about 27% of the best lines could be selected. So we use all this information when making selections. So we use the phenotypic blues as well as the GBVs, so the best lines. So when you select the best lines, we also look at the genomic estimated breeding values. So if a line has good genomic estimated breeding values and also good grain yield phenotypes, we select those lines, and we also discard lines which have poor GBVs as well as poor phenotypes. That's how genomic selection is helping us with our phenotypic selections as well. And we also phenotype traits like grain yield in flat fiber irrigation, and we also do genomic selection on traits like grain yield and flat fiber irrigation, grain yield in bed to irrigation and drip, early heat, grain zinc and iron, stripe blast in Ludhiana, Fusarium head blight, septoria, spot blotch and blast. So all these different traits are important for specific breeding pipelines. So all these traits are not phenotyped in the stage 1 yield trial stage. So we exclusively use genomic selection and the GBVs to make selections for all these different traits. And moving on, so all these GBVs have to be used in a selection index, and that's how the breeders can use them in selections. So we've evaluated different selection indices, and we have the Eigen selection index method, which does not use any economic weights. And then the classic linear phenotypic selection index, or CIMMYT index, where we have to specify economic weights. And for different use cases, we've identified different selection indices to be appropriate. And for, let's say, for selecting for the irrigated pipeline for the grain yield ESIM, we identified the ESIM index to be appropriate. As you can see, these are the correlations between the grain yield phenotypes and the ESIM index. And all these are the correlations between the grain yield genomic estimated breeding values in different environments and the ESIM index. And you can see there's a very high correlation with the true phenotype. And there's also very good correlation of the index with the GBVs in different selection environments. That's exactly what we're looking for. And this helps us to very much in our selection index, in selection decisions. And we also have the genome-wide association mapping results for different traits. And what I'm showing you here, the mapping results for stem rust in this cycle, for stripe rust in this cycle, both of which have been evaluated in Jora, Kenya. And you can see different regions significant for these traits. And we also make use of these significant regions, and we which use this information to understand if this is expected and what new novel regions are present in this cycle. So all of this information is used in our selections. And the next, we move on to the stage 2 yield trials, which have about 1,120 lines. And here we integrate genomic estimated breeding values as selection decisions. And all of these lines in stage 2 have data on grain yield in different environments, iron and zinc in different environments. And the figures here show you the prediction accuracies for grain yield in the bed fiber irrigation, flat fiber irrigation, bed to irrigation, drip, early heat and late heat environments. And you can see that ranged from about 0.3 to 0.5 this cycle. And for iron and zinc, so these two traits are also incorporated in the breeding cycle, these breeding cycles. So we have prediction accuracy ranging from about 0.4 to 0.54 for iron and zinc. And these are just different environments where iron and zinc were phenotyped. So here in the stage 2 yield trials, we use a combination of both phenotypic and genomic selection. And next, in the stage 2 yield trials, we also predict these lines for the target environments, for grain yield in the target environment. So this information on the genomic estimated breeding values for grain yield evaluated in different target environments from different cycles is actually integrated in the selection decision. So for example, let's say TPE1, Ludhiana is in TPE1, and data from both CIMMYT 1920 cycle and 2021 cycle were used to predict the stage 2 yield trial lines that are from Obregon. And the information from several different environments and several different cycles were also used to predict the lines with different target population environments. So this is helping the breeders select lines and to target lines with different target population of environments in South Asia. And again, the eSIM selection index method was used, and we now have TPE-specific target selection environment, population environment-specific grain yield selection indices, which help the breeder in selecting and targeting lines to the TPEs. And then we have the South Asia bred breed genomic production yield trials, which have about 485 lines. And here we integrate both genomic estimated breeding values as well as the phenotypic values and selection decisions. And we have the GEBVs for different sites in the TPEs, Faisalabad in Pakistan, Ludhiana, India, Nowshera, Pakistan, Bhairava, Nepal, Jamalpur, Bangladesh, Pusa and Jabalpur, India, and Sindh in Pakistan. And you can see the prediction accuracies, the within year cross-validation prediction accuracies range between 0.2 and 0.46. And all this information, the GBV is generated, also delivered to national partners. And this information has been used along with phenotypic selection in the selection decisions. And we also do some quantitative genetic analysis. For example, we find genetic correlations between the different sites in South Asia. So this figure shows that the genetic correlations between all these different sites in South Asia, between Jabalpur, TP3, which is a quite different environment compared to all the different TPEs. And you can see Ludhiana, Pusa in TPE 1 and 2 had a high genetic correlation this cycle. So lots of different patterns that we observe, and all this information is fed back into the selection decisions and also in optimizing our target locations, and also in a lot of simulations, work in progress in the CIMMYT breeding program. And to summarize my presentation today, so we've seen a lot of different utilization of a lot of data in the breeding program. So we've used genomic selection, a combination of genomic and phenotypic selection. So the traits that are indicated in the brown color here, all of them are traits that are just selected based on genomic selection and are not phenotyped in these different yield testing stages. So we have exclusively genomic selection for these different traits, and then combination of phenotypic and genomic selection for the other traits. So in total, about 700,000, more than 700,000 genomic estimated breeding values we obtained in the 2021 cycle and utilized in selection decisions. And besides these, we also use selection indices, results from GWAS and quantitative genetics, all of which have helped to the CIMMYT breeding program very much in moving towards a very highly data-driven breeding program. And with that, I would like to acknowledge the Bill and Melinda Gates Foundation, USAID, the Foreign, Commonwealth and Development Office, BGRI, Kansas State, and Cornell Universities for their very good support in the work that I just presented. Thank you very much.